So, split brain research is potentially the most likely 16 marker should biopsychology have a 16 marker in it this year in 2025. So, we're going to go through the key details. This is a simple study, so you need a lot of detail to gather enough marks to get full marks in answering the question, and specifically use of specialist terminology. So for the study, to outline it, obviously it's a standardized study that has been used multiple times on animals and humans to look at specialization of hemispheric function. So the first keyword that you use, and you break down your study into aim, procedure, findings, and conclusions. So starting with the aim is an opportunity to say that it's a study to investigate hemispheric lateralization, and you would explain what that means. Then you move on to the procedure. First of all, we know that the participants had undergone a corpus callosotomy. That means that the corpus callosum had been operated on, meaning that the right and the left hemisphere no longer communicated with each other, and that offered the unique opportunity to study them separately. Carrying on with the procedure, we're going to explain how the activation of only one hemisphere was ensured by asking participants to fix a spot in the middle of the screen, and that means that the stimuli would only go into either the right eye or the left eye. That means that from the right eye it would only communicate to the left hemisphere, and vice versa, meaning anything presenting on the right side of the screen would go to the left hemisphere, anything presented on the left would go to the right. Just a note, there have been some variations that we will mention in AO3 where they asked participants to wear an eye patch to make sure that they would only have information presented on one side of the screen, and they did the two hemispheres separately. Either way, it was to control for the eyes hovering and therefore information going to both hemispheres. The other detail about the procedure that you want to specify is that all images were presented for only a tenth of a second, and that's positive, and we'll see it in the evaluation, both because obviously every image was presented for the exact amount of time to every participant, so it was highly standardized, and also that reduction in time meant it was very difficult for both eyes to catch the image because it was presented for such a short amount of time. And what participants were asked was to identify what they had seen. They might have been presented with words or images on one side of the screen, the other side of the screen, and sometimes on both sides of the screen simultaneously. The findings in Sperry's original that then kind of have been replicated with lots of different stimuli, but objects on the right field, therefore, that went to the left hemisphere could be named, but objects on the left field could not. And that kind of consolidates those findings that we already were aware of, that the language centers seem to be in the left hemisphere. Interestingly, the items that went on the left side and therefore to the right hemisphere were not consciously processed. The people would say that they hadn't seen anything, but they could draw the object with their left hand. That means that the information did go to the right hemisphere, but the participants were not aware of that information being in there. However, they could draw the information, they could also recognize a face from a lineup if it had been presented in the right hemisphere, so on the left side, and they could also pick it out of a grab bag with their left hand. So their tactile functions, their face recognition functions, and their drawing ability seems to be superior in the right hemisphere, therefore using the left hand. Conclusions drawn were that the two hemispheres do specialize in different functions, so it supports hemispheric lateralization. What they found is that the left hemisphere specializes in language and analytical abilities, whereas the right hemisphere specializes in emotions, spatial skills, and music skills. What initially was believed that then subsequently was found to be incorrect is that consciousness only resides in the left hemisphere, because people could only consciously report having seen or experienced things that were presented in their right visual field, therefore processed in the left hemisphere. However, Later on, the conclusion was developed, and what they found was that each hemisphere has its own memories and consciousness, but they're not shared with each other if the corpus callosum is damaged, and that's why whatever was consciously experienced in the right hemisphere couldn't be reported back by the participants because they couldn't communicate it because the right hemisphere doesn't have a language center. So in order to evaluate, we're going to use GRAVE just as a mnemonic, just to help you. When you're evaluating a study, not a theory, you can use the GRAVE acronym to jog your memory to different areas that you can comment on when evaluating a study. Now, the last on ethics in this case is not particularly the strongest point, so you can get away without using. And just a reminder, you don't have to use all 5 components to carry out evaluation. It's just to help you lay out your evaluation. It helps you jog your memory, and try to fill as many points as possible. So we're going to start with generalizability. In terms of generalizability, that means external validity. So we can talk about ecological validity and population validity. So the sample was small, there were only 11 participants, and obviously they had unique neurological conditions, so it has very low population validity. And it is to note that even within that group of people, the degree of damage, the type of epilepsy that they suffered from, all these factors were not consistent. So they weren't even representative of each other or of all other split brain individuals. In terms of ecological validity, again, this task is extremely artificial, not only because it's standardized and in lab settings, but it's not relatable to real life because in real life, even people with split brain don't go around with something that is stopping them from having peripheral vision and restriction of their visual field. So in real life, split brain patients don't really experience the impairments that they experienced in this artificial setup, making it highly unrealistic. On the other hand, the reliability of the research is very high. As we mentioned, the standardized procedures, standardized means the same for everyone. So everybody was in the same environment, controlled setting, the use of restrictions for the visual field, and also the presentation for a tenth of a second of every image. The sequence of the image and the content of the images was all the same for everyone, meaning that consistency was ensured and therefore replicability of the study was very high. In terms of applicability of findings, we need to make a distinction between the real life applicability that is almost inexistent compared to the theoretical applicability of findings. As we mentioned before, in terms of ecological validity, the procedure of cutting the corpus callosum is very rare, and even if it, and when it happens, people don't experience the impairment that was artificially uh, created in the lab settings, meaning that it has almost no real life implications. However, the findings were groundbreaking theoretically in supporting hemispheric lateralization, influencing the fields of psychology, neuroscience, education, and clinical practice. For example, obviously for education, if there's any impairment in the left hemisphere, that can explain any learning difficulty, and therefore it has very strong theoretical applicability and it encourages further research in the field. There's also backing for the kind of evolutionary value of hemispheric lateralization. So it helped consolidate this paradigm in science, proposing that each hemisphere has specific specialized functions. In terms of internal validity, we're talking about control of extraneous variables. And so, for example, there are some extraneous variables that could not be controlled. For example, the disconnection between the hemispheres being greater in one type of participant rather than another, some patients having experienced drug therapy, and also a control group was not really a control group because they had not only they didn't have a split brain, but they didn't have necessarily a history of epileptic seizures. So the control was just irrelevant. Uh, there are also some challenges to the validity of findings because, for example, we have a case study of JW that after the damage in the left hemisphere was able to develop the ability to speak using right hemisphere centers. So that concept of plasticity does challenge the validity of Sperry's findings and questions to what extent these are slightly unrealistic or limited, and there could be much more flexibility to um, brain function. And finally, as I mentioned, ethics is not the strongest point you can make, but there are ways to make it a bit stronger by using specific terminology. For example, if you mention the fact that the study uses a quasi methodology, meaning that the IV is not manipulated, but it's occurring beyond the control of the experimenter, then that obviously makes it more ethical because it wasn't Sperry that decided to carry out the corpus callosotomy that gave the operation. They only measured the outcome of this operation. So that is highly ethical in terms of not harming participants directly, but just studying them. On the other hand, we can question the ability to consent of participants that have undergone quite significant brain surgery and had previous potential brain damage from both the epilepsy and the drug use, meaning that we can't be certain that informed consent was obtained. And that is it.